Yeah, Upasana, it is started.
Namaskar and a very good morning, all of you. On behalf of Indian National Young Academy of Science, myself, Dr. Upasana Ray from CSIR, IICB Kolkata, and Dr. Nishant Chakravarti from IIT Kharagpur, welcome you all to today's session of GYANTEEKA. We are going through a COVID-19 pandemic, and early this year, two of the vaccines received approval for emergency use, and eventually the vaccination drive started in India with dry runs, followed by mass immunizations at priority basis. Skepticism, vaccine myths, and misinformation, however, exist and are discouraging many of us availing these vaccines. As a community of young scientists, we think that it's our duty to spread awareness about the facts and propagate right information. Hence, INYAS recently initiated a new webinar series called as GYANTEEKA. Today, we have Professor Gagandeep Kang as our speaker for the second session of GYANTEEKA series. Now, I invite Dr. Chandrasekhar Sharma, Chair INYAS, to kindly introduce our speaker. Dr. Sharma, please.
Yeah, thanks, Upasana. So, of course, it's my great pleasure to introduce today's speaker. Of course, Professor Gagandeep Kang doesn't need any formal introduction, but for the sake of general viewers on our YouTube, let me formally introduce Professor Kang. So Professor Kang is one of the top biologists in India and worldwide, and she is currently working as a professor of microbiology at the Wellcome Trust Research Laboratory in the Division of Gastrointestinal Sciences at the Christian Medical College, Vellore. Before joining CMC Vellore, Professor Kang served as an executive director of Translational Health Science and Technological Institute, Faridabad. As a virologist, her work in the area of virology, especially in the field of observational, interventional, and mechanistic studies on enteric infection and nutrition is commendable. She is well known for her contributions in the area of interdisciplinary research, studying transmission, development, and prevention of enteric infections and their sequelae in children in India. Professor Kang has immense contribution in building national rotavirus and typhoid surveillance networks and clinical trials of these vaccines. She was awarded the prestigious Infosys Prize in Life Sciences in 2016 for her contributions to understanding the natural history of rotavirus and other infectious diseases. In 2019, she became the first Indian woman to be elected as a Fellow of the Royal Society. She is also the first Indian woman to be elected for the fellowship of the American Academy of Microbiology. With this, I again welcome Dr. Khan to join us, and may I now request her to deliver your talk, Professor Khan.
Thank you very much for the very kind introduction. It's always a pleasure to join with the Young Academy of Sciences, because I think you are doing a phenomenal job of reaching out to people and communicating with them. So I'm going to be talking to you about the value of vaccines, and we will start with the value of vaccines even without COVID-19. But in today's world, you can't get away from COVID-19. So we will discuss that at the end. So this is an outline of my talk. What is the value of vaccines? And I think we don't quite recognize that, what the impact of vaccines might be. And then we'll come to how a vaccine is made before we begin to discuss COVID-19. So vaccines are a best buy. And I think there is a lot of information about vaccines that we are not generally aware of, even as scientists. Currently, it's estimated that vaccines save between 2 and 3 million lives every year. There are a number of diseases for which children receive vaccines as part of national immunization programs. And these vaccines contribute not just to the saving of lives, but also to the prevention of disease and severe disease in particular. Vaccines provide for individual level protection, but they are also public health tools. We have eradicated smallpox, the only human disease to be eradicated so far, though we have eradicated an animal disease, rinderpest, as well. And we are pretty close to containing polio. We have only 2 countries in the world where wild polio virus still circulates. And for many, many other diseases that were killers, we have reduced them to something that can be handled. And we've done all of this using prevention strategies. So vaccines really have an indisputable record as truly outstanding public health tools. Now, the impact of vaccines actually goes far beyond the saving of lives and the prevention of illness. Vaccination is an investment. It's an investment for the individual, but it's also an investment for society because there are wider benefits that accrue across a lifetime. When we think about vaccines, we think about, okay, the vaccine prevented disease, so a person did not get sick. So therefore, we have healthcare cost reductions. And because that person was not sick, they were able to work and were contributing to the economy. But there is a broader perspective on vaccines and the value of vaccines. What can vaccines really do for individuals, for communities, and for societies? And this, you will see, is well beyond health. So if we think about immunization, immunization can lead to more equitable health outcomes. It can interact with other health interventions and further reduce morbidity and mortality, so direct reduction and indirect reduction. It can reduce morbidity and mortality at the individual level. It can also protect populations, what is commonly known as herd immunity, which is in the news these days. This reduction in morbidity and mortality leads to better child survival. That can lead to families having fewer children because more children are surviving, which improves in general the health of women and the health of children. That gives us a demographic dividend. We have more people that are able to work. We have more children that are able to go to school. We have a lower healthcare expenditure, and we therefore have more resources available for spending on other expenditures. So all of this taken together affects the shape of society. It also affects the shape of the economy because you have an increased workforce which is more competent, better educated, and that results in increased productivity. So overall, if we think about what vaccines do, you can see very clearly that vaccines go well beyond the prevention of illness and death alone. They have knock-on effects that really do matter. Now, what does this mean in terms of impact? If we think about a world without vaccines, in the 1700s in Europe alone, which was a relatively small population at that time, about 400,000 people died every year of smallpox. In 1798, Edward Jenner was able to take material from the cowpox lesion of a milkmaid, inoculate it into his gardener's son, and then challenge him with the smallpox virus and show that someone infected with cowpox could be protected from smallpox. By 1800, very quickly, vaccines were being given across Europe and North America. By 1900, smallpox had been eliminated from much of the industrialized world. And by 1975, we had the last natural infection of smallpox. And since the WHO and the world is quite conservative and waits for a few years after to ensure that cases are not going to recur, it was in 1971 that we declared that smallpox was eradicated. So 200 years, and we managed to get rid of a disease. That, of course, made the world much more ambitious. It really promoted the use of vaccines. We had in the late 1970s the start of the expanded program on immunization, and that has led to a reduction in the morbidity and mortality of childhood diseases. We have not been quite as successful with adult immunization programs, and I hope that that is something that will change now with COVID-19. So in terms of how vaccines work, I'll just show you two examples. One is measles, and you can look here at what was happening with measles in the United States. Measles is a very, very transmissible virus. If you've heard of R0, the R0 of measles is somewhere between 13 to 15, so many times that of SARS-CoV-2. And you can see that there were hundreds of thousands of cases of measles in the United States until a vaccine was licensed. And once a vaccine was licensed and began to be used, the numbers dropped very, very rapidly. Till now, measles is only seen in the case of outbreaks, particularly in communities that refuse to vaccinate their children. You can also look at a newly introduced disease where there were cases of meningococcal infection in New Zealand. You can see from 1975 to about 2000, the cases continued to increase. And then when a vaccine was introduced, the cases really crashed after that. So once the vaccine started to be implemented in the program, we achieved disease control. So for both an endemic disease, as well as for a disease that causes outbreaks, it became possible to control infections using vaccines. So what are vaccines and how are vaccines made? Vaccines are essentially a protein, a polysaccharide, a nucleic acid, a living vector, or an infectious agent that is alive or inactivated, that when either injected or administered to the mucosa, induces antibodies and cellular immune responses specific to a pathogen that protect against the pathogen or suppress chronic replication. So the idea of a vaccine is to prevent replication so that disease does not happen. It is also possible for a vaccine to protect not only against disease, but also to protect against infection and to protect against transmission. Now, that does not happen for all vaccines. The main goal of a vaccine is to prevent disease. Now, if we look at the development of vaccines, the smallpox vaccine became available in 1798, 2 years after Jenner's experiments. The rabies vaccine was developed by Louis Pasteur, who used dried out spinal cords of sheep to inactivate the rabies virus so that it would induce an immune response but not cause disease. This was in 1885. We had the BCG vaccine in 1927, and this was made by growing the bacillus in multiple cultures over 200 times in order to attenuate the infectious agent so that it could continue to replicate but would not cause disease. We had the yellow fever vaccine in 1935, and it's really strange. We had the yellow fever vaccine almost before we began to understand what viruses were and how they worked. After World War II, we had a series of vaccines that started to be made. And the reason for making these vaccines is almost 150 years after the first vaccines were made, we acquired new tools. We had a number of vaccines that were made against killed whole organisms, and these include typhoid, cholera, which interestingly were developed so that British troops could work safely in India. And many of these vaccines were developed in the UK and were transferred, manufactured in India so that vaccines could be made available first for British soldiers, and then later these were made available to the local population as well. So if we look at vaccines, we started out with killed organisms or live organisms, and then we began to progress to be able to use components of organisms or new technologies for the development of vaccines. These included our ability to use purified protein or polysaccharides. It included our ability to prepare reassortant viruses that could be used to make vaccines easily. And then we started to have recombinant proteins that could be made so that we had vaccines that were much safer than earlier vaccines. So for example, for hepatitis B, the first vaccines that we had were actually derived from the serum of infected patients. And it was possible that there might have been other infectious viruses in addition to hepatitis B. So the ability to make genetically engineered recombinant hepatitis B protein was a significant advancement in the safety and stability of vaccines. Now, usually when we develop vaccines, we follow a process for making the vaccines. We have a preclinical phase, and then we go into clinical studies. Making vaccines is very expensive and usually takes a very long time. So here you see that you could spend many years in the preclinical phase, and then for the clinical testing phases as well, it takes usually several years to develop a vaccine. Before SARS-CoV-2, the fastest vaccine development that we had ever had was 4 years for developing the mumps vaccine. Now, if we look at the technologies, a lot has changed that has allowed us to develop vaccines fast. I've taken you through the first 150 years where the essential approaches were isolate, inactivate, or attenuate, and then inject the virus. And these were used for a range of bacterial and viral vaccines. We started with Edward Jenner, we had Louis Pasteur, and we had Maurice Hilleman, all of whom used these approaches. And then Hilleman was able to go on and use some new technologies as well. The first new technology we got was cell culture, and once viruses could be grown in cell culture, it became possible to make many more new vaccines. I mentioned that hepatitis B vaccines were safer, and recombinant DNA technology was something that we got in 1980. And then in 1990, we found that we could link sugar molecules to proteins, and that allowed us to produce a much more robust immune response that allowed us also to give vaccines to younger children. Otherwise, if you have a polysaccharide vaccine, that's usually used only in children older than 2 years because younger children don't mount an appropriate immune response unless you have a conjugate vaccine. In 2010, for the first time, it became possible for us to take the sequence of an infectious agent, use that to identify an immunogen, and then test that in preclinical studies and move to clinical studies. So a meningococcal vaccine made by GlaxoSmithKline was the first vaccine that was made using this technology. However, in the last 20 years or so, we have had lots of new technologies that have really exploded vaccine design. And a lot of these are now being used for the pathogen of the moment, SARS-CoV-2. We have structural vaccinology that allows us to identify which proteins are really important. We have the success of mRNA vaccines that have become possible only because of studies that were first done in 2005, which showed us how we could reduce the inflammatory response to RNA. We have, for the first time with Novavax, a vaccine that uses a new adjuvant, matrix M. We have human immune response-driven antigen selection approaches, which are all allowing us to make new vaccines. Now, if we look at phases of vaccine development, they are the preclinical phases where we go into animal safety and immunogenicity, and then the various phases that are done in humans. And the numbers, we start small because vaccines are given to healthy people. So it tends to be a very risk-averse process. You don't want a vaccine that is going to have a lot of side effects. So phase 1 studies are done in 10 to 100 people. Phase 2 studies, which look at dose schedule, safety, and immunogenicity, are done in less than 1,000 people. And phase 3 studies do efficacy and safety and can be much larger. And usually after phase 3 studies, vaccines become licensed, and then you can continue to look at different aspects of the vaccine post-licensure. And these studies can become very, very large. So what do we do in preclinical development? We look at vaccine characteristics, we define the characterization, we develop a manufacturing process. It needs to be something that is very reproducible, and this is checked by the regulators. We establish processes for quality control to make sure that batches are similar. What are the approaches to sterility? How will it be measured? And for particularly inactivated vaccines, you want to make sure that whatever you've got is not capable of replication at all. Then you do efficacy studies. If you have an appropriate animal model, you can do immunogenicity studies. In India, the requirement is 2 small animal species at a minimum for preclinical development. Other regulators have other requirements, and if there are good model systems, those will be required. In phase 1 studies, we look at safety in healthy adult volunteers. You look for the absence of serious or unexpected effects, and this includes the potential for spread. You can do increasing dosage. So for example, the Moderna study tested 3 doses, 15 people with 3 different doses for a total of 45 individuals. The doses can be increased. You can evaluate a single dose schedule, or you can look at several doses. This study usually will not include a control. Sometimes it can be planned as a control study. It really depends on whether this is a disease that has been around before or not. So usually it's less than 50 people who have not been exposed to disease. In phase 2, you go generally into the target population or into a population that is high risk. These here you start to now include controls, and these can be randomized double-blind controlled studies with either an active comparator or a different vaccine or a placebo. And you can have many studies in a phase 2. You can use it for dose ranging, you can use it for schedule, you can use it for association with other vaccines. You can look at response in the presence of existing antibodies. So there are a lot of criteria that are used in phase 2. You look at safety, tolerability, what does the immune response look like, what is the functionality of the antibodies. If you were doing T cell responses, this is where they would be added in. And then you go to phase 3 studies. In phase 1, you establish safety. In phase 2, you really understand the immune response. And in phase 3, you actually try to see, does the vaccine prevent disease or not? You continue to look at safety, and here the evaluation criteria are cases of disease that meet the definition. So when we hear about Covaxin having 43 cases for an interim analysis, these are cases of disease that meet that case definition. Okay, so this is the target population can be very, very large. And if you look to see what is being measured, you can measure many, many things. But the key thing that we are measuring is, does this prevent disease as we defined disease? There are also additional studies that are done, consistency studies, bridging studies. In India, when we get a vaccine that is made overseas to come into India, we do a bridging study, which means that we will do immunogenicity, trying to estimate whether our immune responses are the same as immune responses seen in a population that has efficacy data. Then the interpretation of that is, if immunogenicity is the same, then we expect efficacy to also be similar. That is not always the case, but we hope that it will be. And then once a vaccine is licensed, we continue to look for very rare side effects. And we look at effectiveness of the vaccine. Does the vaccine continue to protect in real life as it did in the clinical trial? And then we look to see how long is the protection maintained. So that's how a vaccine is made. And just to end, I thought I'd walk you through very quickly with where we are with COVID-19 vaccines. We have a number of different platforms. The older platforms are in the top row: live attenuated, inactivated vaccines, virus-like particles, recombinant proteins that are produced in a variety of expression systems, bacteria, insect cells, etc. So those are vaccines for which we have technologies. We have the ability to make them in very large numbers of doses where vaccine manufacturers know what needs to be done. And then on the bottom row, we have the new vaccines. The first licensed vectored vaccine was only licensed in 2019, and that was the vaccine for Ebola. And last year, we had a second vectored vaccine licensed again for Ebola. So these were the first licensed vectored vaccines, and now we have a number of vectors: adenovirus vectors, measles vector, VSV vector being used for SARS-CoV-2. We have DNA vaccines that are in clinical trials. The Zydus Cadila and the Inovio vaccines are in phase 3. We have 2 RNA vaccines that are approved for use, and those are the Moderna and the Pfizer-BioNTech vaccines. And in India, we have Genova that has a phase 1 study for its RNA vaccine. So all of the platforms for SARS-CoV-2 are available with Indian manufacturers, and we have vaccines that are in different stages of development or use. When we started out last year, WHO had prepared a target product profile thinking that SARS-CoV-2 was going to be a very difficult virus to make a vaccine for, because generally it's been quite difficult to make vaccines for respiratory agents. And the target product profile we were looking for was a vaccine that had more than 50% efficacy, that did not use more than 2 doses, was something that would be usable in adults and give protection for at least 6 months. And now looking back, that looks like a really low bar, because almost every vaccine has much, much more than 50% efficacy, and some vaccines are even usable with 1 dose, such as the Janssen vaccine. So we have lots of vaccines that have efficacy data already and are being used in many countries around the world. Now, the worry with vaccines, of course, is the emergence of variants. And why would we need to worry about variants? I think the worry is a real worry, but we have some reassurance from the fact that vaccines work surprisingly well for a mucosal infection. Viruses will always evolve to become more transmissible, and they will evolve to try to escape the immune response. Now, this we have seen when you make neutralizing antibodies to the ancestral version of SARS-CoV-2, it works really well against the existing vaccines, but against the existing, with the existing vaccines. But with the new variants that we have, we see much lower neutralizing antibody responses, particularly with variants that have this E484K mutation. This has been validated by the fact that the trials that have been done in South Africa have shown lower efficacy with the Novavax, Janssen, and AstraZeneca vaccines. Lower efficacy does not mean no efficacy. And I think what we are going to see is that vaccines will have some level of protection against the variants, but we might need to think about whether we want to make another generation of vaccines with the variants so that we have better protection against the variants as well as the ancestral strain. Will we wind up with a multivalent vaccine? It is something that is possible, but we will need to wait and see what the data show us. So I think for SARS-CoV-2, it's very important to remember that we were able to make vaccines in 10 months. We can actually do it in a lot less if we work closely with the regulators. We currently have wave 1 vaccines and wave 2 vaccines, which are vaccines which are much more suitable for national immunization programs that don't have the high level of controlled temperature requirements. Those vaccines are currently in development. So what we want is vaccines that will be cheap and can be given to people very, very widely. And many manufacturers are working on those wave 2 vaccines. Now we need to think about wave 3 vaccines, and clinical trials with Moderna's vaccine, which is designed based on the variant B1351, have already started. So we may have vaccines that are based on platform technologies that could be developed really quickly if we need to use them. It's not clear that we do. We need to get people vaccinated even as we continue with non-pharmaceutical interventions. I think it's very important to remember that we see variants because mutations happen. Mutations happen because the virus is replicated. If we can stop spread of the virus, it won't replicate, it will not be able to mutate, we won't have variants. So if we need to think about vaccines for the future, I think there is a lot that needs to be done. A lot has been done, but I think a lot more is required. We need much more basic virology. The new technologies that are available for vaccine manufacture, I think we are a little bit behind the curve in India. And as young scientists, it's our responsibility to make sure that we step up our game and work on the technologies that are really cutting edge, that will allow us to make better vaccines faster. I think on the public health front, we definitely need to think about better surveillance. The variants have shown us how important molecular epidemiology is, and we need to emphasize that. But it's important to remember that SARS-CoV-2 is not going to be the last pathogen that we will see causing an outbreak. So we do need to make sure that whatever surveillance we set up allows us to pick up new pathogens as well. And then when it comes to clinical research, we've seen a lot of drama in this country with the vaccine trials that have gone on. If we have better quality clinical research, it won't happen. Things will move more smoothly, and we need to establish the structures to do that. We have been incredibly lucky in our country. The disease has not been as bad as it might be. And our vaccine companies are phenomenal manufacturers. So if we are looking to protect the bulk of our population through vaccination, then our manufacturers are really going to save the day. But for the next pandemic, I think it's our responsibility to make the R&D ecosystem ready with the best available technologies, old and new, so that we can make vaccines and make them fast for ourselves and for the world. India is already a key partner in the COVAX facility, which is seeking to supply 2 billion doses of vaccines to the 192 countries that have signed up to the COVAX facility. India has already provided vaccines to 72 countries through Vaccine Maitri and through the COVAX facility. And this kind of looking after ourselves, but also looking after the world with vaccines is something that is critically important for us as a country that already supplies about 60% of all the vaccines that are used in childhood immunization programs around the world. So if we think about vaccination, I'd like to end with the Sustainable Development Goals. Goal 3 is good health and well-being, and we would think that vaccines would only fit under Goal 3. But I will make the argument that vaccines fit under every goal of the Sustainable Development Goals. So just to talk about Goal 1, which is no poverty. So if we had an immunized, healthy population, that would be a productive workforce that leads to strong economies, and strong economies achieve the goal of no poverty. So with that, I'd just like to say thank you for your attention, and I would be very happy to take questions.
Thank you, Professor Kang, for such an informative talk. Your talk involved not only COVID-19 vaccine, but also educated all of us and our viewers about the vaccine biology in general, how they are developed, details about the aspects that are taken care of while performing clinical trials, and a lot more. We will now move on to our question and answer session. Myself and Dr. Nishant Chakravarti will take turns for the questions. So I'll ask two of my questions first from INYAS end. So the first question is, Recently, it was proposed that the booster dose for COVID-19 can be given in 8 to 12 weeks duration. Some vaccination centers mentioned that it increases the efficacy. Can you please comment on the basis for this suggestion?
So when we think about vaccination, some vaccines are given as a single dose, some vaccines are given as 2 doses, some vaccines are given as 3 doses. And then if you have a gap between that primary immunization series, you may want to give a booster at a later date. We know from the immune system that the longer the gap between 2 doses, the more robust the immune response that you produce. Now, why do we pick then a 4-week gap instead of an 8 or 12-week gap? In India, traditionally and globally, traditionally, we have done this because we want to maximize the immune response, and we want to shorten the duration for which the person is at risk of infection. So particularly in the case of children, if we look at routine immunization programs, we give vaccines at 6, 10, and 14 weeks, 1.5, 2.5, 3.5 months. In the West, the same vaccines are given at 2 months, 4 months, and 6 months. The gap is 2 months instead of 4 weeks. And that's because you make a better immune response with longer gaps. But in India, we think that children will have earlier exposure to infectious agents, so we compress that immunization schedule. Coming to SARS-CoV-2 vaccines, it's not true that we need to give all vaccines with a variable gap. We don't know. There is only one vaccine for which we have data on variable gaps, and that is the Covishield vaccine. Not even Covishield, the AstraZeneca version of Covishield, right? And there what we've seen is that immunogenicity and efficacy improve with the longer gap. So in about 20,000 people overall, the vaccine was given at different periods. The original plan was to give the vaccine as a single dose. They found that when they gave a second dose, the immune response increased. So they made the vaccine into a 2-dose vaccine and decided that they would have a short interval. But because there wasn't enough vaccine produced in the clinical trial for the short interval, some people got longer gaps in between the 2 doses. And what they found was that the longer the gap, the better the immunogenicity, the better the efficacy. So the UK's advisory committee, which is the Joint Committee on Vaccines and Immunization, looked at the data, and based on that data, recommended that there be a longer gap between the first and second doses of the AstraZeneca vaccine. They also said that you could have a longer gap with the Pfizer vaccine. Now, in India, we have only evaluated Covishield for immunogenicity, and we have evaluated Covishield with only 1 time interval. So in making the recommendation for a longer interval, we are relying on the data from the UK. This has been reviewed in the UK. It has been reviewed by the WHO, and the WHO's recommendation is 8 to 12 weeks. In India, our recommendation is for longer than the original 4-week gap, but it's not as far as the WHO's 8 to 12-week gap.
Okay. The next question is, what was an observed hesitance? There was an observed hesitancy among the respondents towards Covaxin because of its admission while clinical trials was on. So it was approved under clinical trial mode. do we have some data? We do have some data now. And what would be your suggestion to the people who are offered Covaxin? Because a lot of people, they don't want to avail this vaccine because of that reason.
I would say take the vaccine. So Covaxin's results are good. We have an interim analysis that is showing 80% efficacy. 80% is pretty good compared to all other vaccines. There's absolutely no reason not to take the vaccine. The problem is when it was approved in January, it was approved in clinical trial mode. And there is no such thing as clinical trial mode. If you ask a clinical researcher, what is clinical trial mode? Either you're doing a clinical trial or you're not. There's no mode. Okay. So I think that was the issue. The fact that every other vaccine, except from Russia and China, were approved only on the basis of efficacy data. Even if it was an emergency use authorization, Pfizer, Moderna, AstraZeneca were all approved only on efficacy data. So I think in India, we had good signals that Covaxin was going to work, but a good signal and data are two different things. So phase 1 and phase 2 is not equal to phase 3. Anyway, we are all past that now. It's clear, the data are good. We should take the vaccine.
Thank you, Professor Kang. I now hand over this session to Dr. Chakravarti.
Thank you, Dr. Ray. And thank you, Professor Kang. It has been an exceptional talk and very informative for everyone. So I think most of the viewers are listening to the term efficacy quite often on various platforms, including yours. So we have some viewers on YouTube who have been asking about this question. For example, Mr. Gautam Mandal is asking regarding how efficacy is exactly measured, if you can comment on that. And I would also like to add one more question, which is similar to this. So based on the efficacy data that is being shown, none of the vaccines are 100% efficacious, which is quite understandable. So in that case, whether one can even think that they are actually getting vaccinated, and if yes, then how long can we expect these vaccines to be working?
Thank you. So let's take the first question. How do you measure efficacy? When you design a clinical trial, you design a clinical trial to say, does the vaccine work? And you have to define, what do you mean by is the vaccine working? Usually in vaccine trials, in a phase 3 trial, you're looking for prevention of disease, which requires that you have to define disease. How will you measure disease? Now, for all of the clinical trials that have been done, RT-PCR test positivity is a requirement. But after that, things get a little bit muddy. So not all clinical trials are measuring the same thing. Some of them are measuring fever and a PCR positive. Some of them are measuring fever, another symptom, and PCR positive. Some of them are measuring fever, another symptom that lasts for at least 48 hours, and PCR positive. So you can see that when you say we are measuring efficacy, all trials are not exactly comparable because each trial has a different definition of how it is calling a case in the trial. So what we do in the clinical trial to measure efficacy is we compare the numbers of cases, however we define them, in people who received vaccine and people who did not receive vaccine. Many of the trials will do a 1-to-1 split. So half the people get vaccine, half the people get saline injection or a different vaccine. Some trials do a 2-to-1 split where 2 people get vaccine and 1 person gets the placebo or a control vaccine. But essentially what we are trying to do is measure rates in people who received vaccine and people who did not receive vaccine. So if you had 1,000 people who were vaccinated and 1,000 people who were not vaccinated, then and among the unvaccinated, 100 people developed disease, became cases as we have defined them, RT-PCR plus fever and another symptom. Then you look to see how many cases did you see in the vaccinated. Suppose in the 1,000 vaccinated people, you saw only 10. Then the vaccine has 90% efficacy because you reduced the number of cases in the vaccinated by 90%. Okay, so that was your first question. After
Yes.
that, how long will vaccines work? I wish I knew. So based on infection, we think that the protection lasts for months. How many months? Will this months turn into years? I think this is something that we will have to wait and see. Among vaccinated people, how long does it take for their immune response to wane? How long does it take for them to get reinfected? Now, we haven't had the virus around long enough to tell us that you will have years of protection because the virus hasn't been there that long, but it's looking pretty good. The rates of reinfection that we are seeing are very low. And when people get reinfected, it tends to be very mild infection. Then the other question was about, no vaccine is 100% safe. No vaccine is 100% efficacious. We as humans are too variable to allow that to happen. We will always have some minor side effect. We will always have somebody who will have no protection from infection. We are heterogeneous and we need to recognize that. So if we had a correlate of protection, a test that we could do and say, this person is protected from disease and that person is not protected from disease, that would be easy, right? You get the vaccine, you get the test, and now I can go party anywhere I want. Or, I need to be careful. I need to think about taking a booster or a different vaccine. At the moment, we don't have that correlate of protection. We are working on one, but it'll take some time before we have that. So we do need to continue to be careful once we have vaccines. But I think the important thing to remember is that while individually we might not know whether we ourselves are protected or not, vaccinating the population as a whole will reduce disease and transmission. So even not being protected individually, as soon as vaccination ramps up, we will see life being a little more normal.
Thank you, Professor Kang. And I think your answer also answers another question which was leading, which was like, with the given choices, what would you suggest, vaccine or a mask and social distancing? So I think both of them are needed, as you have already explained. So I'll take on the next question, which says that we know mutation is common. Mutations are common, and the spike proteins which get mutated act as the major antigens for most of the vaccines that have been under development. So because these mutations are a continuous process, so in the long run, would you be suggesting or would you be thinking that the whole virus-based vaccine would be a better option to ensure epitope diversity? Can you comment on that, please?
So why would you want to have, when you know that the spike protein is responsible for generation of neutralizing antibodies, you know that pure spike-based vaccines are working very well, 95% efficacy, 90% efficacy with some of the vectored vaccines. They're working very well. Do you need all the other antigens? Will they give you better protection than the best, right? So I could see that
Yes.
a whole virus or an inactivated virus might have some components that would be partially protective, but I don't see how they could be better than. I could see a combination schedule as well, where you try to use one dose of an inactivated or a live attenuated vaccine with one dose of a spike vaccine. I think there is going to be a lot more immunological studies that we will need to do in the future to get the optimal vaccination schedule and the optimal vaccines.
Okay, thank you. Thank you very much. So that brings me to my next question. And I think this is a question that even I have in mind. So as we agree that there are a lot of asymptomatic cases of people around us, so do you think it is rational to do an antibody testing for people who are willing to get it done prior to taking the vaccine?
So from an academic point of view, it makes sense. You know, if you know your antibody status, good for you. But from a public health point of view, if you can't do it for everybody, why bother? You're going to give them the vaccine in any case, right? I actually think that where it would be most interesting would be to study immune responses in people who are seropositive when they receive their first vaccine, and then follow them up for a long period of time, because my prediction would be that they would have the most robust protection from infection, a combination of natural infection and vaccination.
Thank you. I'll take up one more question from our YouTube viewers. So Mr. Amog Dighe mentioned that in the principle, and you have discussed about this, that during the course of developing these vaccines, so several vaccine dosing schedules are discussed and looked into. So he assumes that a few candidate those schedules are selected and compared. So can you give us an insight into how these schedules are finally selected?
So I started with, well, I did mention during the talk that Moderna's phase 1 trial compared 3 different doses of mRNA in 15 people each. And they found that the highest dose caused a lot of reactogenicity. The lowest dose induced a good immune response, but the middle dose produced an even better immune response. So they decided to balance the immunogenicity and the side effects of the vaccine to pick the middle dose. And that's what they are using. It turns out that Moderna's middle dose is actually many times the amount of mRNA that is in the Pfizer vaccine. So it's possible that Moderna could get a good immune response by diluting its dose further, but then that would make it a different vaccine. So I think Moderna is planning to try that out with the next set of trials that it's doing. But this kind of dose ranging study is usually done several times during vaccine development. For other vaccines, you might do a series of dose ranging studies. In the case of SARS-CoV-2, because we were in a hurry, there were fewer dose ranging studies, and that's why we might need to go back and revisit in the future for some of these vaccines.
Sure. Thank you, ma'am. So you also mentioned about the Covishield and the AstraZeneca variant version of the Covishield. So could you let us know what, or is there any difference?
Compositions are identical. This was technology transfer from AstraZeneca to Serum Institute of India. They have been evaluated both in terms of the compositions of the vaccines, but also in terms of immunogenicity of vaccines using the same tests, and the vaccines are producing identical immunogenicity. So it's the same thing made in India. instead of made at AstraZeneca's other factories outside.
Okay, thank you, ma'am. I think there are lots of questions that are still flowing in, but I don't think we will have enough time to cover all of them. I'll just pick up one last question, which is, I think, of interest for everyone. So we do know that the post-vaccination symptoms vary among individuals. So is it due to the viral component or the adjuvants, or solely dependent on an individual's immune response? And with this, I will pass it on to Upasana after this. Thank you.
I think, given the fact that the vaccine is the same in many people, the response is different among those many people. Who do you think it's because of? It's our individual responses. We respond differently to infections. I think everybody knows somebody who will, whenever there is a cold going around, that person will get it. In our families, we'll have some people who always get cold, some people who never get cold. Similarly, people who have diarrheas or fevers. It is our individual variation that makes us susceptible to illness, to side effects. It's not because the vaccines are different. We react to components in the vaccines.
Thank you, ma'am. So I'll hand it over to Dr. Ray.
Thank you, Nishant. And thank you, Professor Kang, for your talk as well as such an engaging discussion. I'm sure our audience have enjoyed listening to you. Those who will listen to your talk later from INYAS's YouTube channel will also get benefited. So thank you once again. Thanks to Chair INYAS and thanks to the entire GYANTEEKA team. We also have Dr. Veda Krishnan and Dr. Rajiv Dev here controlling other aspects of this session. So thank you all. INYAS will come back with another talk, the third talk of GYANTEEKA series soon. With that, stay safe, stay healthy, and Jai Hind.
Thank you, Professor Khan. Thank you. We can stop the live.